I'm very pleased to be here, and I really, as the previous speakers say, want to thank the organizers for putting together such a great group. I'm learning quite a bit myself, so I really am pleased to be here. What I was charged with was the idea to look at cumulative risk. And so what I thought I would do is try to talk a little bit about integrating temporal and systems-based models for detecting cumulative impacts, and to share with you some of the lessons from the Pacific Northwest Children's Health Research Center that I direct in the state of Washington. To do that, what I want to do is to then outline the talk as follows. Well, what are life stage and systems-based approaches? And we've heard a little bit about that, so I can probably skip a few slides. I think I have a slightly different emphasis on this. Two, how have we applied these approaches to look at cumulative exposure and risk? And again, we're looking at agricultural practices and agricultural communities. So I'm going to give a little brief description of our cohort and descriptions of findings that will place this framing in context, and then to talk a little bit about what new framing needs we need, what new framing areas we need for our results and analysis. So hang on to your seats, we're going to go forward. When we talk about life stage, we're talking about the different areas that you've heard about so extensively this morning. And putting that then as a fit into the risk assessment paradigm has always been a little bit awkward, and it's especially awkward when you then add on the end, after outcomes, a need to look at populations. And so when you think about populations across life stage, that's extremely important, and it's really where the rubber hits the road and what we've been talking about, how it's difficult to get there. So rather than making this more simple, I'm going to say that I actually think we need to be a little bit more methodical and a little bit more quantitative in how we take these assessments, because I think sometimes we end up taking samples and biomarkers and we don't actually have the framework yet that we need to know when and how frequently we need to take these markers. So I was surprised when Martin said, well, we already know that we have 7 to 8 stages that we need to take a sample in. Well, I actually think that one of the things that's a challenge there is that we don't have enough duplicate samples from individuals to understand within-person variability from one day to the next. And you'll see for the examples that I'm going to give here with pesticides that are not, in general, persistent, I think it provides a good example of looking at what we need to be able to answer the types of risk questions that we have and be able to go on to cumulative risk. I'm going to do some of this framing in the context of what the National Children's Study has been trying to do and the questions that they've had. For several of you, I know, have been sitting and listening in, maybe on the IOM conference call on Friday and so forth, to look at how they prioritize exposures and health outcomes, and it's exactly the kinds of things that we're talking about here. We want to have a variety of priority exposures. We want to look at taking those from a variety of endpoints, but we also have priority health outcomes, some of which that overlap and some that don't. And then if you throw in that there's a cumulative impact, then we have all the more challenge in trying to do that kind of an evaluation. One thing that they got extraordinarily right was the idea that they broadened the definition of environment. And as you see here, physical, chemical, biological, genetics, and psychosocial are exactly what we're saying that we need to have and to have as baseline information and going forward. The challenges that they've put out, and one of the purposes of the IOM meeting was to determine how best to get that information and what the balance would be for biological measurements, environmental measurements, how many measurements do you need to take at the community level, household level, individual level, and how many samples do you need to take across life stage, and supplement it with questionnaire. Now, you've heard hints about some of the complexities of interpreting this, but really this is exactly what they're trying to do, and I'm going to try to address where I think our children's health study started to address some of these points. To do this, one of the things they did was to realize is that across disciplines, whether you're a physiologist, a developmental biologist, or a neonatologist, or EPA, that you have different definitions of life stage. And these are defined both in terms of the types of activities you're involved in, the eating behaviors, the eating patterns, but also in terms of what things your discipline is interested in. So, for example, EPA laid out very good models for this, and the WHO guidance on principles for children took that and expanded a bit on that. And then if you've listened again, the issue is, well, one sample during pregnancy, does that answer for first, second, and third trimester? Well, if you're definitely a physiologist and a developmental biologist, you know that the answer is no. So we've been fortunate enough to be funded both by the US EPA, NIEHS, and NICHD to leverage some of the center cohort to look more specifically at these questions. I'm going to talk about three areas. I'm going to talk about exposure response measures, mechanisms of toxicity and non-chemical stressors, and genetic susceptibility, and to use the Children's Health Center cohort to answer some of those questions. I'm not going to talk about the neurobehavioral studies that are going on as well. To do that, I'm going to have to recap for some of you that have heard me talk about the Children's Health Study recently, so bear with me. I've got 10 slides here, and I think we'll get to that. This study was conducted in eastern Washington in the area of Yakima County. Here it's shown in green. If you look at this, I'm going to point at my computer here, not too helpful here. It's one of the areas of highest fruit production in terms of, well, we're one of the highest international producers of pome fruit, in particular apples. And as you can see, it's also correlated quite well with the highest amounts of organophosphates that are used in our area. This just shows the geographic construct on this. These are some associated with cities, some townships, some labor camps, and also proximal to Indian reservations for the Yakima Reservation. So, and it covers several county areas besides Yakima, including Benton County, which is also one of the high areas of OP usage. To do this, I also just need to remind you, ASA did a great job of talking about the need for looking at organophosphates and understanding how these break down. They break down in the body and also preformed entities. The parent compound here, chlorpyrifos, can break down into diethylthiophosphates. It can also form oxone, which is of interest because this is the one that's associated in particular with the acetylcholinesterase inhibition, and also form DEP. It can also form specific metabolites such as TCP related to chlorpyrifos and methylchlorpyrifos. So what we've done in our study is to look at the parent compound in the blood, to look at the formations of the specific dialkyls, and also to then look at the enzymes that are associated with these different pathways. Cytochrome P450s, you'll see, and there's a myriad of them, are associated both with the formation of the oxone, toxic in this case, as well as the breakdown of the compound to presumably less toxic moieties here. Enzymes such as PON peroxidase 1 are associated with the breakdown of the oxone, and there's been collaborative studies between the University of Washington and University of California, Berkeley, with Asa and Brenda and Nina Holland to look at the age of onset and genetic variability and abundance of the PON1, and we'll get to that in a minute or two. So this is true for other organophosphates, but there are important distinctions. And the differences are that there are different proportions of the cytochrome P450s that are involved in the metabolism, different amounts of the different dialkyl phosphates that are formed. In this case, with azine phosmethyl, a similarity is the oxone is again still believed to be associated with the acetylcholinesterase inhibition, but in this particular case, you form DMT and DMTP. And one of the questions that we were able to answer, and I'll mention, is that PON1 is not involved in this, and that had not been known before. It had been speculated, but we have the first human evidence to show that that is indeed the case. So let's go forward. As Asa very nicely showed, that there's a portfolio of various diethyls and dimethyls, and what we have done is tried to look at is how much of that complexity can actually start to inform you about multiple exposures and timing of exposure and attributable risk to different exposure pathways. So to do this, what we've done is to actually use a framework, and this is a very simple biomarker framework where we looked at both particularly pathways of exposure and community-based pathways. We looked at child, we looked at adult, we looked at household-based assessments, and then we looked at a community-level assessments, and then we looked at markers of early biological response, such as acetylcholinesterase, and factors such as genetic susceptibility and molecular markers to get to early alterations in this study. So these are a series of cohorts. We have 800 individuals enrolled by Betty Thompson into this Yakima Valley cohort. It's been used for a variety of evaluations, including diet and nutrition and cancer. But of particular interest for what we've been doing is to actually look at associations with community interventions that reduce pesticide exposures, as well as then to look at very specific agricultural practices and seasons, and to look at a household basis with a matched parent and child of farm worker and non-farm worker families to look at this over time. And so these studies have been going on for over a decade. And of particular interest today that I'm going to get to is talking about a series of nested studies that they're then used to look at some specifics about exposure, specifics about mold, specifics about cortisol and non-stressor pathways, real-time metabonomic exposures, and I'm not going to talk so much about the respiratory health studies, but Catherine Carr is here, and she can tell you she's waiting for results as we speak here. Okay, so what lessons can we learn from this community and its children? As we showed early on, is that most children were exposed, multiple exposures to the dialkyls, and multiple exposures to different diethyl and dimethyl metabolites. We also had the situation where in our state, the intervention for education is at the applicator standpoint. Applicators were trained, and their households and their children actually had lower levels of OPs than what did our thinners. Thinners were not covered under our statutes for specific additional information and education, and in those cases, the thinner children and adults had higher levels of exposure. And if we compare these to NHANES, the adults in this cohort are, the average is tenfold higher than the national NHANES studies. And part of that is due to the fact that we looked at and sampled at specific seasons of peak use versus other seasons of use. This was done largely by building off the work early on that Gloria Coronado did, and Ilda, who was killed in a tragic car accident this last year, and Betty Thompson on this. So this just shows orders of magnitude concentration for the distributions of farm workers and non-farm workers. And you can see here there's a dramatic shift in this, and this is for concentrations of the dimethyl thiophosphate. And in this case shows a similar shift, not to the same magnitude, but again, the same shift for the farm worker children. So the issues are what contributes to this variability and how much can we pick up. So this slide shows for the dimethyl phosphate metabolites, and what it shows is that for just looking at these metabolites, and this is plotted against then 3 different metabolites here. I'm going to focus just on one here for a moment. And this is looking at thinning season and off-season or non-spray season. Remember, our scenario is a little bit different. We only produce apples one time a year, and so we have very controlled activities that go on. Thinning is when you workers go out and actually pick and cull the buds so that you get an optimal crop, and it turns out it takes place at specific times, and specific times for reentry after organophosphate usage in this case. But what you see very dramatically here is that for the farm workers versus non-farm workers across the 3 days of sampling, and these were done day 1, day 3, and day 5 in this thinning season, there's a consistency of signal for all 3 metabolites, and there's a consistency in the thinning season associated with the farm worker that you don't see with the non-spray seasons, nor with the non-farm workers here. So it's a pretty nice pattern, and when you look at this, or not nice, you remember I'm a toxicologist, so I have a jaded view of what's nice, but it really starts to lay out specific times of exposure and the implications of exposure pathways. In this case, is for the children, again, statistically different in the farm worker versus non-farm worker, and it's associated and consistent across the 1-3 days of thinning. So how do you start to put this together? This is just one pesticide, one compound, and so one of the things that we get asked all the time is, oh, all of these compounds are preformed, it could be there and so forth, so how do you work on it? Well, we had the luxury because of Dana Barr that the relationship here is shown now that we have measurements for the azine-phosmethyl, and this was not true for chlorpyrifos or any of the 4 other parent compounds here. And this has shown that you actually can relate the amount of AZM in the blood with the actual amounts of the metabolites. The strongest is the DMTP for a variety of reasons, but it's all correlated and it's highly significant. So that allowed us then to go forward and look at acetylcholinesterase inhibition with parent compound across these times. And so when you do that, we were able to develop relationships in this cohort that acetylcholinesterase inhibition increased 15% per 100 micrograms of DMTP per liter in the urine. And this is all controlled because we had both spray and non-spray seasons, so each person served as their own control. But it allowed us to then go on now and start to interpret how much acetylcholinesterase inhibition is related to DMTP and to be able to interpret our results in a more broad-based manner. Now, I said this was held up true for azine-phosmethyl, but not for CP. So let's look at the nested studies and see what else these individuals are exposed to at the same time. So one of the things is actually mold. And this is collected both and dust, and how much of it is in dust. This is one of the things that we were doing was also evaluating different dust collection methods to make sure that they're representative. I'm not going to talk so much about that, but what I am going to do is to talk about the results. So for azine-phosmethyl, phosmet, malthione, and chlorpyrifos, the amounts of OP concentrations in the Housedust persisted between the thinning and now I've got 3 seasons in here because we broke down thinning, harvest, and off-season on this. And again, the relationship between thinning and off-season and off-spray, no spray, holds up. But it also shows that the concentration in the Housedust persists for longer than just the period of thinning. So here we have significant differences, again, between farm worker and non-farm worker families, and it gives us a hint at the persistence. When you put, just so you have some reference here, when you put the values for azine-phosmethyl in the dust in these households alone, these values start to push about 20 to 30% of the population of adults and about 10% of the children over the limit just for a single compound, not for any of the other considerations here. So when you put it into a cumulative, these are concentrations that are of concern, and especially because they're consistent there. So mold, what do? Well, we used 2 factors. We used pyrosequencing, which is the 454 method to actually sequence the exact species of fungi that you have in the dust, as well as then looked at the ERMI, environmental related moldiness index in this. And we special thanks to Steve Vesper at EPA. And the reason that we used this relative moldiness index was the idea that this had been in other studies associated with an increased odds ratio with as a predictor for asthma. We don't know if it's a predictor for asthma in our study yet, but we do know that this range of exposures of ERMI scores was included a -5 to about a 14, which is a sort of normal range, but we haven't laid this together with knowing if that's now related with our respiratory outcomes, but that will happen on that. And that will be then crossed with the pesticide exposure. The pyrosequencing data was interesting. It was probably the first time that they had picked up forest signals. And of course, this is interest because of the cedar dust and cedar issues of mold. So it's probably indicative of the geographic construct for these individuals, and also a potential indicator these people may be working in forestry, which also uses some of the OP. So we have to investigate that further. But it shows the idea of that you can use these new genomic tools to actually get a characterization of these kinds of mold products and start to then look at this. All right, so if we come back, what did we see in genetic susceptibility? Well, this is important because I mentioned before the life stage, not all exposures are the same, and it's because of the temporal onset of the metabolizing enzymes that are present there. So we had previously published with the Chamaco study that the onset of PON1 was age-dependent and so forth, but what we hadn't shown was that the relationship of PON1 had nothing, it did not relate to the acetylcholinesterase inhibition. So that's important because normally we classify sensitive and non-sensitive individuals based on PON1 sensitivity, and that is not true for this biomarker of response for ACE and phosphomethyl. So although we think about these organophosphates as being and having a common mode of action, even the metabolism and the genetics associated with them pose challenges. The next slide was then showing that we also used the DMET chips, which look at 225 genes. This is not quite the whole genome sequencing, but it handles about 2,000 drug metabolism markers that are important for predicting response for drugs. And in this particular study, one of the interesting observations that had not been shown before in humans is that the cytochrome P450-3A5 did have significantly different slopes for inhibition of acetylcholinesterase, and that then allows us to now understand a new factor in genetic susceptibility that goes across the different organophosphate pesticides. So by putting the genetic information in context of an early biomarker of effect, and the early biomarker effect being the common biomarker of effect that goes across these pesticides, we can start to look at how we might compile information that informs a biomarker-specific approach or systems approach for risk assessment, in this case, for one type of class of compounds. The other thing is that we were interested in looking at, we had been asked to look at a variety of markers, biomarkers, for cortisol, and to put that into the context of two types of questionnaires that can be used in these kinds of cohorts. So Rosalind did a beautiful job this morning of talking about why social stressors are relevant to children's health, and we used and cited this here. What we did here was for the NCS is to collect hair, blood, saliva, and urine samples for looking at long-term cortisol. This is in our non-pregnant women, but the mothers in this study. And then to look at the sort of most cost-effective and acceptable measures that would relate to two types of questionnaires. Betty Thompson many years ago had produced a Hispanic-specific stress questionnaire, and so we're using that, and we're also using the generalized stress questionnaire that's a variation that WHO uses for stress and well-being, and we're making comparisons. And in this particular case, it's before and after, and again, we're using the hair. In this case, it's not trimester, but it's at the start and at the end of our study, which was a 6-month study in this particular case. To do this, and then the other thing that we'd link this with was this metabonomic approach that Martin was talking about. So if you take urine samples and you collect it in a kind of virtual biobank and come back and say, could you pick out cortisol signal from that? Can you see cortisol, and how does that vary by households? So we use this in addition to this. And sure enough, we're able to pull this out. This is what the metabonomic, and don't panic, the slides don't get more complicated than this. But this is what the metabonomic bioinformatics looks like from what Martin talked about earlier. But the idea is in this particular case, because we match parent and child, we match household, and we match day 1, day 3, and day 5 measures, we're able, and then match it with these 4 other biomarkers of physical biomarkers and then questionnaire, we're able to start to sort out patterns in this. This is just purely the pattern based on households, so you can start to see the patterns there. So these are households for adults, and these are and child, and then they are linked through ANOVA to look at a subset of metabolites. So then, having these results, we then did a cassette approach and pulled out cortisol with standards. And it turns out that, and these are very preliminary results, that the most amount of variability that we have is actually in our non-spray season. There are a variety of reasons why this might be the case, and I can't interpret it completely, but it starts to look at and gives you an idea that we'll look at seasonal household, child, adult, and different factors of stress in this community. Just an example, this is the biokinetic model that Marissa Smith has just presented on the phone webinar the other day, and at one meeting thus far. It's not quite published yet, but what this does is allow us to use a kinetic approach, like a pharmacokinetic approach, and relate the biomarkers from all of these sources and get a cumulative impact score so that we aren't just looking at one marker, but it's actually a way to relate the biomarkers together. So that's been exciting because we're very pharmacokinetic and dynamic oriented in the center. This is just an example of the kinds of questions that come up in the Hispanic specific questionnaire for stress. And we're using the one with Snipes and Thompson et al. So what I've hopefully done is to show you in these particular studies is that we were looking at exposure, we're looking at responses across various levels of biological observation, in some cases biochemical or genetic. In some cases, we're actually looking at a response that's occurring at an individual level. And we're putting this together for the time stages that we're looking at here, but hopefully constructing a model that allows us to look more broadly. So why? We've been talking about this in the concept of getting this into regulations. I chose to look at the regulations for OP pesticides for cumulative. And despite the disparaging comments about a cumulative mechanism of action, I do agree that this is limiting. But I also want to remind you that outside of OHIA, there have been very few cases where we have actually looked at even combining the effects of different chemicals with the same mode of action. So I think there are lessons to be learned from this, and we could perhaps use this as a jumping point to look more closely. So I remind you that this is within a structure of both single chemical, but it's under FQPA, and that we actually combine these for common modes of action. We need a lot of help on this because our studies are suggesting that that's not sufficient, as are suggested by others. But this idea that we would be looking at this by defining common endpoints of impact or common interaction patterns, I think, is still usable, and we need to think about it as we move forward in this, because this might be a baby step in getting to this broader concept of looking at cumulative risk and making an impact down at the bottom where we really look at populations that differ dramatically. We do feel that you need a quantitation, and this has been emphasized by others, the idea that we need metrics that allow us to distinguish between different states. And we think that the within and between person variability is very high in these studies for agents like pesticides and for issues in this community for stress. So it's a very dynamic community. Lots of things happen, and we all have the stories about loss of individuals, criminal behavior, et cetera, that is piled on top of this. So key findings, there's a complexity to environmental exposures, and therefore, by implication, a complexity to the outcomes that would be anticipated from this. I think we should embrace the complexity. We actually felt that we got a lot more information about sources of exposure by embracing the complexity rather than reducing the evaluations down to simple reductionist measures. We also believe that this still is a need for longitudinal and life stage considerations. I don't believe that one sample during pregnancy answers all of this. And if we're going to take two samples, take them a day apart or even hours apart to get at the within-person variability and repeatability. I'm going to stop on this and say that this is a slide that I've used before at the end, but there's no better voice than from the community. In this, the children get the story here. This is after a series of presentations to grade school classes. The students were invited to enter a contest. This woman won this contest, and the reason she won is that here she is in the red dress. She tells her husband to wash his clothes separately and to keep his clothes and boots outside the door. She's going dancing. So I think what better can we say than that it's a cumulative effect. It has to be the parent and the child, and it has to be done at household levels as well as individuals for intervention. So again, thanks to many people. I have the pleasure of standing up here and talking about this, but there were many graduate students and my colleague Betty. We wouldn't have been able to do this. And the funding sources, EPA, NIHS, and NICHD really have allowed us to explore on this cohort. Thank you.